The Molecular Vision of Life, Caltech, the Rockefeller Foundation, and the Rise of the New Biology. By Lily E. K. Introduction. During the 1930s, a new biology came into being that by the late 1950s was to endow scientists with unprecedented power over life. These 3 decades culminated in the elucidation of the self-replicating mechanisms of DNA and an explanation of its action in terms of information coding, representations that laid the cognitive foundations for genetic engineering. Scientists could now manipulate genes on the most fundamental level and attempt to control the course of biological and social evolution. They laid claim to the secret of life. The new biology, which became known as molecular biology, emerged as a dominant disciplinary trend. Its molecular vision of life promised to function as Occam's razor, paring down the convoluted explanations offered by traditional biological fields. This new science did not just evolve by natural selection of randomly distributed disciplinary variants, nor did it ascend solely through the compelling power of its ideas and its leaders. Rather, the rise of the new biology was an expression of the systematic cooperative efforts of America's scientific establishment, scientists and their patrons, to direct the study of animate phenomena along selected paths toward a shared vision of science and society. The aim of this book is to understand the historical process that propelled molecular biology to its dominant disciplinary status by uncovering the motivations and mechanisms empowering its ascent. It does so by focusing on 2 key institutions, the Rockefeller Foundation and the California Institute of Technology. As has been well documented, the Rockefeller Foundation served as the principal patron of molecular biology from the 1930s to the 1950s. Caltech, a primary site for implementing the Foundation's project, became the most influential international center for research and training in molecular biology. Why did the Rockefeller Foundation launch and sustain with massive support a new biology program at that moment in history? From the entire range of contemporary biological interests, why did scientists and their patrons privilege and promote a molecular study of life? Why was Caltech selected as a primary site, and what accounted for its remarkable influence? Studied together, the trails leading to the answers reveal a synergy between intellectual capital and economic resources, a potent convergence of scientific goals and social agendas, shaped initially by the cultural imperatives of the interwar period and later modified by the experience of World War II. Some preliminary directions would be helpful before embarking on these trails. Specifically, a clarification of terms and meanings accompanies the delineation of the constitutive elements of the primary argument. Molecular biology, a new biology? The term molecular biology and the authentic novelty of the field have been a subject of some debate among scientists and historians. Coined in 1938 by Warren Weaver, the director of the Rockefeller Foundation's Natural Science Division, the term was intended to capture the essence of the Foundation's program, its emphasis on the ultimate minuteness of biological entities. Point 2. However, if by molecular biology one means the program sponsored by the Rockefeller Foundation, this definition would include many practitioners of life science, example, biophysics and immunochemistry, not typically associated with the term. Current parlance tends to equate molecular biology with DNA molecular genetics, a definition that would exclude most of the research in life science during the 1930s and 1940s. To complicate matters further, it appears that some scientists who would never have identified themselves as molecular biologists were retroactively, in light of the high status of molecular biology during the 1960s, all too eager to reconstruct their careers as part of the molecular biology success story. Others, especially biochemists, resisted the sweeping of their parent disciplines into the whirlwind of hybridization. In light of these divergent meanings of the term molecular biology, it is necessary to outline a set of criteria that together explain and justify use of the term in this study. Even though separately some of these structural features of molecular biology were not novel, assembled and amplified in a single program, they eventually did constitute a coherent intellectual and institutional framework that departed sharply from traditional modes of biological research. 1. Programmatic statements about the newness of the new biology were not limited to its patrons. The new biology, as geneticist Thomas Hunt Morgan put it in 1928, stressed the unity of life phenomena common to all organisms rather than the diversity. Thus, the new biology would concentrate, for example, on respiration or on reproduction as a central biological, in contradistinction to biochemical, problem, regardless of whether the object of study was a mammal or a bacterium. 2. Based on this rationale, it became far more convenient to study fundamental vital phenomena on their minimalist levels. Thus, the new biology increasingly employed simple biological systems, primarily bacteria and viruses, as phenomenological probes or as conceptual models, the line of thinking that led to Jacques Monod's notorious dictum that what is true for the bacterium is true for the elephant. 3. By cleaving life processes from their host organisms, the molecular biology program aimed to discover general physico-chemical laws governing vital phenomena. In so doing, it distanced its concerns from emergent properties, from interactive processes occurring within higher organisms, between organisms, example, symbiosis, and between organisms and their environments, thus bracketing out of biological discourse a broad range of phenomena generally subsumed under the term life. Concomitantly, this physico-chemical approach bypassed historical explanations in biology and developmental and evolutionary accounts of life. Processes: the arrow of time. The new biology generally acknowledged only mechanisms of upward causation, ignoring the explanatory role of downward causation. 4. It would borrow methods not only from physics, mathematics, and chemistry but also from other fields of life science: genetics, embryology, physiology, immunology, microbiology. The new biology aimed to transcend disciplinary boundaries and employ whatever tools the problem at hand demanded. Although the transfer of techniques between fields was certainly not new, the design of a large-scale program based on interdisciplinary research encompassing several disciplines was unprecedented. 5. By defining life in terms of fundamental physico-chemical mechanisms, molecular biology ultimately narrowed its principal focus to macromolecules, and until the mid-1950s it meant primarily the giant protein molecules. Molecular biology was based on the protein paradigm, the premise that the salient features of life, reproduction, growth, neural function, immunity, could be explained through the structures and functions of proteins. In fact, guided by the protein paradigm, research on antibodies occupied a key position within the new biology. This important chapter, however, has been written out of the history of molecular biology. 6. Molecular biology thus defined the locus of life phenomena principally at the submicroscopic region between 10⁻⁸ and 10⁷ centimeters. That this region was the main functional domain of the new biology had immense consequences for the form and content of research. 7. This domain could be investigated primarily with complex and sophisticated apparatus, specifically designed to investigate life at this range of dimensions. Whereas only a couple of decades earlier biology laboratories housed mainly microscopes, petri dishes, and autoclaves, the new biology laboratories displayed an imposing technological landscape. Electron microscopes, ultracentrifuges, electrophoresis, spectroscopy, X-ray diffraction, isotopes, and scintillation counters became the sine qua non of biological research. 8. The cognitive focus on the molecular level also shaped the social structure of research. Partly because of the interdisciplinary nature of the problems to be investigated and partly because of the complexities and costs of the new apparatus and the intricacies of the techniques, research problems were often defined by the instruments designed to examine them and were increasingly addressed as team projects. Molecular biology studies thus entailed structural changes in the organization of departments and laboratories and the prizing of cooperation as an institutional strategy and a personal ethos. One important disciplinary consequence of these characteristics of molecular biology was the loosening of the traditional grip of medicine over biological research. Whereas in the past biological research was shaped by the extent of its service role to medical schools, and, to a lesser extent, agriculture, the new biology, which focused on fundamental physico-chemical explanations, microorganisms, and submicroscopic processes, had only indirect links to medicine. 5. As it turned out, the medical connection did affect the growth of molecular biology, even at Caltech, but in a convoluted and variable manner. During the 1930s, however, the designers of molecular biology carved for it a spacious, medicine-free niche within the disciplinary ecology of life science. They intended to create a new science of life, a science whose tributaries eventually converged on the molecular study of the gene. Rockefeller Foundation, Knowledge and Cultural Hegemony. The cognitive and structural reconfigurations of molecular biology were greatly facilitated through the powerful resource base of the Rockefeller Foundation. During the years 1932 to 1959, the foundation poured about $25 million into the molecular biology program in the United States, more than one-fourth of the foundation's total spending for the biological sciences outside of medicine, including, from the early 1940s on, enormous sums for agriculture. Although there was a great deal of fluctuation in the annual expenditures for science during that 25-year period, the pre-World War II level of Rockefeller Foundation support for molecular biology amounted on average to about 2% of the entire federal budget for scientific research and development. This figure gains significance when we consider that the lion's share of government support for the life sciences went to agricultural research. When we also include the indirect effects of the Foundation's support for molecular biology in Europe and its massive support for biomedical research, the financial resources for molecular biology become even more impressive. It is clear that the Rockefeller Foundation was in a strong position to shape fields in life science that fell outside the scientific domain of the federal government. 6. The Foundation's power to shape life science transcended the dollar amount of its investment. Its effectiveness lay in creating and promoting institutional mechanisms of interdisciplinary cooperation through extensive systems of grants and fellowships, and in systematically fostering a project-oriented, technology-based biology. As the correspondence files, reports, and diaries of the officers reveal, during the 1930-1950s the Rockefeller projects became densely interwoven with the scientific agenda of those universities that were heavily supported through the Foundation's molecular biology program. The Foundation's network permeated their academic infrastructures. A significant number of Rockefeller trustees held top administrative positions in the universities. The Foundation officers cultivated advisors and contacts in nearly every discipline and had detailed knowledge of the academic traffic. The officers became central to what in effect became an informal peer review system. It was not uncommon for scientists and administrators to consult the Foundation's grapevine regarding academic appointments, reputations, personalities, travel, and potential projects. Weaver was even invited to sit in on faculty meetings, and within the limits of professional discretion and philanthropic etiquette, the officers were pleased to be closely involved on many interlocking levels of the scientific enterprise. One of the strongest motives for redirecting academic practice and for creating institutional mechanisms compatible with the Foundation's design was the drive toward cooperation, a key feature in the molecular biology program. Cooperation did not mean just collaboration, the grassroots, spontaneous activity of scientists sharing theoretical and experimental tools. Interdisciplinary cooperation, with its emphasis on group projects, was a long-range strategy and an overarching philosophy. Inaugurated in 1933, the molecular biology program was planned as part of a joint venture between the divisions of natural, medical, and social sciences. Within the Natural Science Division, the molecular biology program itself was cooperative on several levels, among biological disciplines and between biology and the physical sciences. Again and again, as one reads through the Rockefeller reports, one is struck by the constant references to various projects as the isotope group, the protein group, or the neurospora group. One is also struck by the special attention to what the Foundation referred to as cooperative individualists, men whose intellectual enterprise included a managerial temperament. The term cooperation had an even broader meaning. It was more than an institutional strategy for fostering interdisciplinarity. As a modification of the extremes of laissez-faire, cooperation was a political and economic ideology of the evolving corporate structures of post-World War I America, specifically science, industry, and business. The reorganization of biology around the standard of cooperation reflected the broader reorganization of American science during the Great War and the restructuring of social relations in corporate America. Mirroring its industrial and business sponsors, the new scientific enterprise no longer extolled the virtuosity of the individual. Just as the multi-unit business structures depended on the team player and the coordinating manager, so the new science relied on management and group projects directed toward interdisciplinary cooperation. There was a remarkable commensurability between levels, between the goals of the new program and the institutional structures that supported it, between ideology and form. Similarly, the infusion of massive and sophisticated apparatus into biology must also be understood on several levels of significance. Instruments are not mere devices for discovering objective reality but complex processes of intervention for representing nature, processes that alter nearly all aspects of scientific practice. The new technologies not only raised the cost of research, they brought about structural changes. With large-scale commercial equipment almost unknown before World War II, new instruments often had to be constructed in situ. Workshops and special rooms had to be built to house the new equipment, thereby greatly expanding not only the budget but also the physical space of biological laboratories. These technologies also demanded a type of technical expertise that allied biology more closely with the physical sciences and engineering. This alliance too was part of the Foundation's goal. More fundamentally, the reification of the molecular level as the essential locus of life, with the attendant reorientation of laboratory practice, altered the epistemological foundations of biological research, making the representation of life contingent upon technological intervention. Conceived at the twilight of an era characterized by its faith in technology and business, the design of the new biology not only reflected the particular bias of its principal architects, physicist Max Mason and mathematician Warren Weaver, but the more general bias of a technocratic elite who had dominated American culture during the 1920s. Simon Flexner, Rockefeller Foundation trustee and director of the Rockefeller Institute for Medical Research, in 1934 disapproved of the new program. Not only did he question the soundness of a biology managed by physicists, he doubted that a collection of instruments and techniques constituted a new biology. In ways that Flexner had not envisioned, the program did just that. The force of the Foundation's molecular biology program, and especially the effective management of Warren Weaver, have been amply acknowledged and debated. Caltech geneticist and Nobel laureate George Wells Beadle observed that during the dozen years following 1953, the elucidation of DNA structure, Nobel Prizes were awarded to 18 scholars for research into the molecular biology of the gene, and all but one were either fully or partially sponsored by the Rockefeller Foundation under Weaver's guidance. Not only did Weaver help shape Beadle's scientific path, he influenced the careers of hundreds of others, inside and outside the field of molecular biology. Historians of science have offered divergent interpretations of the pervasive influence of the molecular biology program and Weaver's influential role. They have ranged from celebration to condemnation. The program was fruitful and innovative, the program was elitist and conservative, merely a process of technology transfer, it was a subversion of biological knowledge and of the academic order. All have concurred, however, that the program did have a profound impact on life science. Although several works have examined the structures, mechanisms, and effects of the Foundation's molecular biology program, only scant attention has been paid to the broader intellectual and social agenda within which the program was nested. Little has been said about the inscribed cultural and ideological premises or the historical forces underlying the development of a molecular framework for the study of animate phenomena. This book addresses this lacuna by situating the molecular biology program within the Rockefeller Foundation's Science of Man agenda, thereby viewing the program as both a scientific and a cultural enterprise. The motivation behind the enormous investment in the new agenda was to develop the human sciences as a comprehensive explanatory and applied framework of social control grounded in the natural, medical, and social sciences. Conceived during the late 1920s, the new agenda was articulated in terms of the contemporary technocratic discourse of human engineering, aiming toward an endpoint of restructuring human relations in congruence with the social framework of industrial capitalism. The support for life science must be seen within that larger investment in the human sciences. Within that agenda, the new biology, originally named psychobiology, was erected on the bedrock of the physical sciences in order to rigorously explain and eventually control the fundamental mechanisms governing human behavior, placing a particularly strong emphasis on heredity. This conjunction of cognitive and social goals had a strong historical connection to eugenics, to its promise and perils. By 1930, the Rockefeller Foundation had supported a number of eugenically directed projects. By the time of the inauguration of the new Science of Man, however, the goal of social control through selective breeding had suffered severe setbacks. As an intellectual program, eugenics guided by the crude principles of Charles B. Davenport had lost much of its force, and as a social movement it carried the stigma of racial prejudice and political propaganda. Eugenics as such became a scientific liability. The quest for rationalized human reproduction, however, never quite lost its intuitive appeal, even when it was later modified by the Nazi experience. For the architects and champions of a science-based technological utopianism, human engineering through controlled breeding remained a compelling social vision. Thus, one of the sub-arguments of this book is that eugenic goals played a significant role in the conception and design of the molecular biology program, an argument that hinges on the politics of meaning. Precisely because the old eugenics had lost its scientific validity, a space was created for a new program that promised to place the study of human heredity and behavior on rigorous grounds. A concerted physico-chemical attack on the gene was initiated at the moment in history when it became unacceptable to advocate social control based on crude eugenic principles and outmoded racial theories. The molecular biology program, through the study of simple biological systems and the analyses of protein structure, promised a surer, albeit much slower, way toward social planning based on sounder principles of eugenic selection. Time was seldom a deterrent for the visionaries of the Rockefeller Foundation. As Wycliffe Rose, head of the International Education Board, used to remind his pragmatic colleagues, remember we are not in a hurry. Moreover, as Raymond Fosdick, trustee and later president of the Rockefeller Foundation, acknowledged during the 1920s, in the quest for enlightened social control, there is no royal road to the millennium, no shortcut to the promised land. Indeed, support of the human sciences by the Rockefeller philanthropies did not begin during the early 1930s with their Science of Man agenda. By then they had supported biology for about 15 years and had been the main force behind the development of the social sciences in America. Scholars have generally concurred that the Rockefeller philanthropies have played a leading role in shaping the human sciences, and that their projects were in large measure an effort to build a base of technical expertise in order to lay a rational foundation for social reform. However, the meanings and interpretations of the Foundation's motives and its impact have diverged. The political significance of such terms as technical expertise, rational foundation, and social reform have been contested, and studies of the motivations behind the wholesale promotion of specific intellectual programs and institutional structures span a wide spectrum of approaches. They range from Marxian analyses to liberal perspectives, from arguments of economic determinism to claims of intellectual autonomy. Point 11 Circumventing these interpretive dichotomies, this book examines the directed autonomy of leading practitioners of molecular biology sponsored by the Rockefeller program. We shall see how the social goals of the private sector, as individuals and collectively, interacted with the research goals of these life scientists in a mutually enhancing manner, as a nuanced process of consensus formation. The reference to the private sector encompasses industry, foundations, and individual donors, usually of industrial fortunes. My analysis does not place the Rockefeller philanthropies outside the ideological framework of the business ventures that gave rise to them. The corporate structure of the philanthropic enterprise mirrored the structure of the business corporation, and the visions of the Foundation's trustees, leaders of business and industry, reflected their ideologies and social world. True, legal distinctions such as tax laws and non-profit status did differentiate these quasi-public institutions from their parent corporations, but they are proximate mechanistic causes. Programmatic statements, reports, and memoranda from the various Rockefeller divisions attest that on social and ideological levels there was no fundamental separation of purpose between the heads of corporations and the leadership of the Foundation. Drawn primarily from the business sector, the Rockefeller trustees exerted their ideological and economic influence on general policies and on specific grants. Trustees' approval of academic projects was a continuous concern for the Foundation's officers. Point 12 Animated by a potent conjunction of Protestant values and technocratic visions, the Foundation's civic missions were formulated within the dominant cultural categories of race, class, and gender, as well as within a socio-economic framework that defined norm and deviance for individuals and groups. The Rockefeller philanthropies cultivated scientific and managerial elites in order to address the root causes of social dysfunction, culturally specific and historically contingent forms of maladjustment. Their projects aim to restructure human relations and to develop social technologies commensurate with the material and ideological imperatives of industrial capitalism. These observations are not Machiavellian attributions or pronouncements of academic subversion and co-optation. The complex set of relations of scientists to patrons and of intellectual programs to the social agenda can be better explained within an analytic framework of cultural hegemony, through the explicit and tacit constitutive processes of consensus formation. Within that framework, power includes intellectual, cultural, political, and economic power, and mental life is not a mere shadow of material life. From this perspective, the maintenance of hegemony does not require active commitment by an academic constituency or by subordinates to legitimate elite rule. Rather, the two reinforce each other in a circular manner to form a hegemonic bloc sustained by formal and informal systems of incentives and power sharing, particularly through half-conscious modes of complicity. Hence, this viewpoint does not regard hegemony as a form of subtle coercion or top-down social control, but, rather, as an interactive process between different social groups vying for power. 13 Within this theoretical framework, it does not matter that many of the scientists funded through the Rockefeller Foundation's molecular biology program were unaware of their social function within the science of man agenda. As we shall see, the leaders of American life science, many of them acting as scientific advisors to the foundation, did understand the larger picture, but even they did not always share all the goals of their patrons. The rise of the new biology was a process of consensus building among interdependent though not identical professional constituencies with common as well as separate goals. In search of patronage, however, most American leaders of pure science did argue for their service role. Their pleas and pledges often projected conflicting images and contradictory purposes: pure research as disinterested knowledge, research as investment in economic growth, human betterment, and political power, objectivity as a mark of professionalization, relevance as a measure of social worth, science as a democratic institution, science as an elite enterprise. Perhaps no scientific figure had embodied and reinforced these contradictions more pointedly than the German émigré Jacques Loeb, America's emblem of pure Wissenschaft, who, while cleansing biology from the taint of pill-pushing, articulated his intellectual mission in terms of the technological control of life. 14 Thus consensus can form without active complicity. Whether practitioners of molecular biology valued their works as contributions to a broad social agenda, or through perceived intellectual autonomy they deployed the appropriate rhetoric to fund their basic research, is secondary to the process of consensus formation. As the evidence shows, however, at the highest levels of management there was a substantial degree of agreement. As members of a national elite with similar social, religious, and educational backgrounds, the leaders of science and the foundation's officers and trustees were cut from the same cultural cloth. This resonance facilitated the emergence of a hegemonic bloc. What is of primary importance, however, is that through their authority scientists did empower the Rockefeller program. By offering expertise, they supplied an instrumental rationality that not only legitimated their own enterprise but also validated the cultural objectives of their patrons. California Institute of Technology, Engineering and Consensus. No single institution exemplified this cultural resonance between patronage and science better than the California Institute of Technology. No other academic enterprise so clearly expressed the powerful conjunction of intellectual capital, economic resources, and institutional structures. The selection of an exemplary institution affords an opportunity for a detailed examination of these trends and operations, a close-up view that would be obstructed by a wider perspective on the rise of molecular biology. At the same time, there is a limit to extrapolating from the experience of one institution. Within these polarities of density and diffusion, the focus on Caltech represents a strategic optimization between the textures gained from a microstudy and the generality lost without macroanalysis, because on several counts Caltech serves as a paradigmatic case study of the rise of molecular biology. In a single center, we can fruitfully examine the development of some of the major cognitive trends, the focus on physicochemical genetics, the emphasis on instruments, and the dominance of the protein paradigm, with its heavy emphasis on immunology. We are able to follow institutional and social mechanisms in action, group projects, and interdisciplinary cooperation. To be sure, there were some hurdles and conflicts, but we generally witnessed the process of consensus formation on social goals, representations of life, and the future shape of biological research. A cursory glance at Caltech during the 1930-1950s reveals that the institute nurtured some of the most important founding fathers of American molecular biology. All supported by the Rockefeller Foundation, these future Nobel laureates built at Caltech influential research schools based on the cooperative model. The premier geneticist Thomas Hunt Morgan was engaged in 1928 to head the new biology division, and not only because of his Mendelian virtuosity on Drosophila. No stranger to business fortunes and foundations politics, he was sought for his effective scientific management, which had propelled genetics from a disciplinary marginality to the vanguard. In his cooperative new division, comprising genetics, biochemistry, biophysics, embryology, and physiology, researchers were expected not only to build bridges between these biological fields but to forge links with physics and chemistry. The German physicist Max Planck formed some of the earliest links between genetics, physics, and mathematics, establishing at Caltech during the late 1930s the foundations of the phage school. This research program, which employed bacterial viruses as conceptual models of gene action, has been generally recognized as one of the most fruitful approaches to the gene problem and a principal turning point in the history of molecular biology. His intellectual leaps did not fully account for his ascendancy, however. When he received a professorship in biology at Caltech in 1946, it was only partly for his personal scientific output. His strength lay in having established a socially cohesive program and a collaborative network spanning several disciplines, institutions, and countries. A similar vignette emerges around George W. Beadle. A brilliant geneticist in his own right and founder of American biochemical genetics, he demonstrated his interdisciplinary flexibility early in his career at Caltech. Moving from corn to fruit flies to fungi, by the early 1940s he was managing an impressive neurospore project at Stanford. Groomed for leadership by the Rockefeller Foundation and by Caltech's establishment, Beadle returned to head the biology division after World War II with a track record of cooperative projects spanning university, industry, and the military. The career of Linus Pauling displays a similar pattern. Director of Caltech's chemistry division and one of the principal architects of molecular biology, Pauling towered as both scientist and manager. His studies of the chemical bond and of protein structure revolutionized concepts of the architecture of living matter and were central to America's prominence in X-ray crystallography. Propelled by Dionysian forces far stronger than any of his colleagues, Pauling's intellectual ambition was reinforced by bold managerial maneuvers that placed him and Caltech at the forefront of Rockefeller support and of the production of molecular knowledge. These research schools, in turn, attracted during the 1930s to 1950s visiting scholars and collaborators from around the world. The 1930s to the mid-1940s were challenging times for Caltech's program, but the mid-1940s were a watershed. Delbrück predicted in 1947 that Caltech in the coming years will be to biology what Manchester was to physics in the 1910s. 15 Indeed, like Manchester, Caltech became an international training ground, a nursery for a new science. Nearly every leading molecular biologist by the 1960s had had some connection to Caltech. Hundreds of young life scientists, graduate students, postdoctoral fellows, research associates, passed through the Kirchhoff and Gates laboratories during the formative period of molecular biology, most of them supported by Rockefeller money. Propagating the new techniques and research strategies, this second generation spread the molecular vision of life to other research and teaching centers. Caltech, of course, was not the sole center for the new biology. The developments at Caltech, however, become even more remarkable from a comparative perspective when viewed in light of the Rockefeller Foundation support of molecular biology at other universities. A survey of the Foundation's annual reports from 1930 to 1955 reveals that the Foundation supported molecular biology projects in scores of elite institutions but invested the largest sums in 6. In accord with its long-standing policy of supporting the strong, making the peaks higher, the University of Chicago, Caltech, Stanford, Columbia, Harvard, and the University of Wisconsin most consistently received grants for various projects in molecular biology. Of these institutions, the Foundation regarded Chicago and Caltech each receiving about $5 million, and Stanford about $1 million, as, in that order, the most promising centers for developing a unified program of molecular biology. Among these three, Chicago presented the most convincing case in this country and the most important program opportunity. 16 Columbia, about $1 million, Harvard, and Wisconsin, less than $500,000, occupied favorite positions within the Rockefeller academic network, but these pockets of excellence did not evolve into coherent programs in molecular biology. Why did these institutions lag so far behind Caltech? To understand these developments, we must not only count dollars, grants, after all, were a vote of confidence, but see the molecular biology projects within the ecology of knowledge, in terms of the goodness of fit between cognitive activities and their institutional contexts. From this perspective, the Wisconsin case is quite easy to explain. The university did enjoy special status. Both Weaver and Foundation President Max Mason spent most of their academic lives there. Fields central to the molecular biology program did thrive at Wisconsin, especially biochemistry and biophysics, endocrinology, microbiology, and genetics. The dominant influence of the food and drug industries, however, tended to guide research projects in these fields primarily toward agricultural, veterinary, and medical processes and products. Harvard's case is straightforward as well. Biology at Harvard had always lived in the shadow of medicine. By 1940, with genetics at Harvard nearly extinct, the principal projects in physicochemical biology were conducted primarily in the medical school. In both places, Wisconsin and Harvard, a new biology could hardly develop an identity independent of its traditional service roles. 17 The Columbia situation is somewhat more challenging to explain, as the university made significant contributions to molecular biology. In addition to work in protein chemistry, there was promising research under the direction of physicist Harold C. Urey on the biological effects of heavy hydrogen, the work on radioactive isotopes by Rudolf Schunheimer, and of course Erwin Chargaff's groundbreaking studies of nucleic acids composition. This research, however, was conducted mainly in the biochemistry department at Columbia's College of Physicians and Surgeons, with little interaction with the zoology department or with the perspectives of genetics. Under the direction of geneticist L. C. Dunn, the zoology department, formerly towering with the stature of Morgan and E. B. Wilson, emerged from a state of partial eclipse only during the early 1940s. Zoology possessed neither the will nor the capacity for interdisciplinary partnership with the medical school. Rockefeller grants from the mid-1940s to the mid-1950s to the zoology department aimed to restore Columbia's eminence by supporting primarily mammalian and population genetics. These institutional and intellectual dynamics were unfavorable for fostering a unified molecular biology program. 18 At Stanford, though a relatively young institution, two trends had solidified by 1930, the strong presence of the medical school and the lack of interdisciplinary cooperation. Through the trustees' directives and the presidency of Ray L. Wilbur, former dean of the medical school and later Rockefeller Foundation trustee, medicine came to occupy a dominant fiscal and disciplinary position at Stanford. The School of Biological Sciences was medically oriented, including departments of anatomy, bacteriology, physiology, botany, and zoology, and the Hopkins Marine Laboratory, but until Beadle's appointment in 1937 there was no research in genetics. Interesting projects developed at Stanford: ultracentrifugation studies of macromolecules, research of bioelectric phenomena, radiation, developmental mechanics, bacterial chemistry, cell metabolism, cell biology, and viruses, and during the early 1940s biochemical genetics. For structural and personal reasons there was little common planning and collaboration. In fact, the lack of cooperation between chemistry and biology was one of the motives behind Beadle's departure in 1946. The strong medical influence and the sluggish response to the calls for cooperation were detrimental to the emergence of a unified interdisciplinary program with a distinctive biological signature. 19 The University of Chicago offers the most fruitful comparative perspective on Caltech and the other four universities. Its institutional network was densely intertwined with the Rockefeller hierarchy, and the foundation's support of Chicago equaled that of Caltech. Through the energetic leadership of Frank R. Lilly, advisor to the foundation and director of the Marine Biological Laboratory at Woods Hole, the Chicago program during the early 1930s exemplified both the tradition and the vanguard of American biology. In fact, Lilly played a decisive role in shaping the intellectual design of the molecular biology program. With a large group working on the biology of sex, with luminaries such as Sewell Wright in genetics, F. C. Koch in biochemistry, Carl Ashley in psychobiology, and later biologist Paul Weiss, Chicago's life sciences were at their peak. Given the remarkable strength of the social sciences and the strong interest in psychiatry at the medical school, the University of Chicago should have rightly emerged as the premier center for implementing a program that sought to address the genetic, developmental, and chemical processes of behavior in cooperation with the social and medical sciences. Great traditions do have costs, however. They are grounded in history and tend to impede alternative paths. Were regarded as preclinical fields. Lilly's directorship had sustained the division's distinctive biological identity, but with the replacement of Lilly as dean of the Division of Biology by immunologist W. H. Taliaferro, the medical school came to have greater influence on biology during the 1940s, tending to orient it toward clinical research. Furthermore, Chicago had a strong tradition of evolutionary biology and natural history nested within the university's formidable intellectual framework of progressive evolution. The evolutionary standpoint was fundamentally incompatible with the mechanistic conception of life. That Chicago's genetics had a strong focus on populations and that under the leadership of Paul Weiss biological research addressed developmental and interactive processes from organismic and holistic perspectives attested to the viability of older traditions at Chicago, traditions that competed with the new disciplinary goals during the formative years of the molecular biology program. 20 Caltech had none of these competing disciplinary traditions, no evolutionary biology or natural history, no agricultural mandate, and above all no medical school. Despite intermittent pressures from the 1920s to the early 1950s to add medical research to the life sciences, Morgan's original plan to resist the medical imperative prevailed. Plant physiology and agricultural interests experienced similar tensions. Similarly, the strong presence of Theodosius Dobzhansky during the 1930s did not orient the division toward evolutionary biology. The new biology was implanted into a disciplinary matrix that diverged sharply from that of the other institutions. Caltech's biology program explicitly aimed to depart from established biological traditions in order to create a new science of life based on cooperation with the physical sciences and engineering. The engineering setting would be especially compatible with a technology-based biology. In terms of curriculum, new linkages were formed on the undergraduate and graduate levels, producing after a decade a generation of biologists trained in the physical sciences. This redirection of biology was accomplished through Caltech's unique institutional structure. Unlike older universities, Caltech had already distinguished itself during the mid-1920s as an elite institution that championed new fields grounded in interdisciplinary cooperation. The appropriate institutional mechanisms were created from the start in order to encourage a problem-oriented approach to science, structures that were guided by corporate models of management and inspired by the cooperative projects of World War I. Although geographically peripheral and an academic neophyte, by the late 1920s Caltech formed the hub of America's scientific establishment. Its illustrious leadership glided smoothly through the corridors of power that linked academe with industry and the philanthropic foundations. These leaders also forged a formidable alliance with Southern California's business elite. Their scientific and ideological commitment to the region's industrialization situated Caltech squarely within the local political economy, thereby garnering enormous community support, which in turn stimulated the flow from Foundation's coffers. This convergence of institutional and cognitive strategies in the absence of competing biological traditions, buttressed by enormous social and economic resources, explains why, of all the well-endowed universities, Caltech emerged as the premier center for molecular biology. At Caltech we witness a remarkable resonance of means and ends between scientists and their patrons. The late 1930s, a large number of prominent life scientists at Chicago reached retirement age. The nature of new appointments would hinge on administrative reshufflings. The Division of Biology had been integrated into the medical curriculum for 3 decades at that point, and its biochemistry, physiology, pharmacology, bacteriology, anatomy, and psychology departments' formation of a consensus on scientific and social goals based on the primacy of the molecular vision of life. Molecular vision of life. Now that the constitutive elements of the argument are explained, we are in a position to extract the levels of significance embedded in the primary thesis, the molecular vision of life. Above all, we can explore the linkages between the form and content of biological knowledge and the directionality and modality of seeking knowledge. Viewed for the moment strictly on the cognitive level of science, the question arises whether life is molecular or it is only the vision that is molecular. By studying macromolecules, do we study the salient attributes of life, or only a molecular representation of life, one of many possible representations of animate nature? To put it differently, the title of this book indirectly addresses the epistemological tension between various knowledge claims competing for the appropriation of the secret of life, interpretations that, in turn, give rise to divergent social and medical prescriptions. Ultimately, the resolution of this tension hinges on the currently unfashionable question, what is life? A centuries-old query that, although by no means intellectually bankrupt, has proved too challenging for philosophers and is deemed unproductive by most practitioners of life science. My own point of departure is that there are multiple biological realities within which life is systematically explained. Various research schools in life science during either different or the same historical periods have privileged different scientific representations of life, with representations referring to the totality of biological practices that include the choice of biological system, methods, theoretical frameworks, and interpretations of results. Each program has labored under the conviction that its own representational practice grasps the essence of life. Thus, evolutionary biology, in addressing the quintessential property of living entities, their ability to evolve, has given primacy to time and environment as principal arbiters of the structures and functions of organisms, species, and populations, and more recently biomolecules. Various schools of ecology, though differing in their approaches, would agree on the premise that some of the most important processes subsumed under the term life cannot be properly understood outside the interactive models that account for profound reciprocal changes in organisms and their environments. On the organismic level, the followers of Walter Cannon, for example, would give primacy to homeostasis as a salient attribute of organismic life, thereby stressing holistic or integrative approaches to vital processes. Practitioners in these diverse biological fields might value molecular explanations but would surely disagree that molecular representations derived primarily from microorganisms supply an overarching explanatory framework for animate phenomena. The disagreement has deep historical roots. Since the Aristotelian beginnings of the debate of teleology versus mechanism, the question of how much we can learn about life by examining only its building blocks has not been resolved. Mechanisms of upward causation have been remarkably effective for a finite range of biological phenomena but have not effectively explained some of the primary characteristics of life. Neither the Cartesian program, the cell theory, the supposed victory of mechanism over vitalism around 1850, which was actually a confrontation between mechanism and teleology, nor the DNA double helix have successfully accounted for emergent properties of life such as differentiation, growth, evolution, or human consciousness. On the other hand, even for the subcellular level, mechanisms of downward causation have revealed properties and processes of cellular components that are not manifested in their unassembled state. Few would contest that the packaging of viruses or the hardware of bacteria offers only minimal insights into the biological organization of mammals. The abundance of rigorous quantitative anti-reductionist models that have developed during the second half of the 20th century attests to the limits of the mechanistic and physicochemical approach for solving problems of biological organization. Point 21 This broad spectrum of biological fields and the multiplicity of biological realities grounded in divergent conceptions of life make it clear that a number of viable biological programs existed that could have been singled out and promoted by the Rockefeller Foundation during the 1930s. The evolutionary, ecological, and organismic standpoints spotlighted many secrets of life to be unraveled. They supplied different kinds of knowledge about the human body and mind as well as alternative paths to understanding social and environmental maladjustments. In short, there were different possible human sciences. Why then did the Rockefeller Foundation's Science of Man agenda privilege a molecular vision of life? The answer to this question is embedded in the matrix that linked the particular forms of social control sought by that agenda with the specific kinds of control supplied by the new biology. The control of animate and inanimate nature, of course, was not a 20th century project. Viewed diachronically, the interlocking of representing and intervening was inaugurated with the Baconian program, with the birth of an autonomous experimental tradition whose primary aim was to manipulate and control nature for the utility of man and to collapse the dichotomy of the natural and the artificial. 22 From a synchronic perspective, the particular political, economic, and social configurations in early 20th century America gave this conjunction of knowing and doing historically specific meanings. Greatly influenced by Jacques Loeb's project, which had adopted the engineering standpoint toward the control of life, the Rockefeller Foundation officers and their scientific advisors sought to develop a mechanistic biology as the central element of a new science of man whose goal was social engineering. In the social sciences, the emphasis on controlling human behavior gave rise to mechanistic conceptions of behavior, personality, and socialization. Interventionist strategies were inscribed into these scientific formulations. The life sciences aim to map the pathways in the molecular labyrinth of the human soma and psyche in order to control biological destiny. A biology governed by faith in technology and in the ultimate power of upward causation is far more amenable to strategies of control than a science of downward causation, where elements cannot be fully understood apart from the whole. There is seductive empowerment in a scientific ideology in which the complexities of the highest levels can be fully controlled by mastering the simplicity of the lowest. The rise of molecular biology, then, represented the selection and promotion of a particular kind of science, one whose form and content best fitted with the wider, dominating patterns of knowing and doing. The molecular vision of life was an optimal match between technocratic visions of human engineering and representations of life grounded in technological intervention, a resonance between scientific imagination and social vision. The organization of this book is primarily chronological, the narrative interweaving between projects of the biology and chemistry divisions. The first chapter traces the origins of the science of social control, positioning the conception and design of the new biology within the Rockefeller philanthropy's promotion of the human sciences during the early part of the 20th century. The second chapter focuses on the emergence of life science at Caltech, situating the new biology within the Rockefeller Foundation's program and within Southern California's social nexus and political economy. Chapter 3 explores the intellectual and institutional dynamics in Morgan's new biology division, with a particular focus on the ecology of the new biological knowledge. Chapter 3 is followed by an interlude, which traces Caltech's commitment to the protein paradigm back to the intellectual trends that had dominated genetics and biochemistry since the beginning of the 20th century. The interlude provides the necessary background for subsequent chapters, especially Chapter 4. Chapter 4 explores some of the influential American researches in physiological genetics, that of Jack Schultz, George Beadle and Boris Afrasi, and Max Delbrück. It focuses on the linkages originating at Caltech between genetics and embryology, biochemistry, and biophysics, which contributed to the primacy of proteins in the nascent molecular biology program. Chapter 5 traces the emergence of the chemistry division under Pauling as a major center for molecular biology, a joint effort between Caltech, the Rockefeller Foundation, and Pasadena's business community. Chapter 6 examines the development of immunochemistry under Pauling as a key feature of the protein paradigm and a decisive advantage for war-related research. Chapter 7 reconstructs the intellectual and social history of Beadle's Neurospora program during World War II, activities that later took him to Caltech as head of the biology division. A second interlude surveys some of the effects of the dislocations of the war on the policies of the Rockefeller Foundation and the impact these shifts in power had on the molecular biology program at Caltech. The final chapter examines the consolidation of the protein-based molecular biology program under the leadership of Beadle and Pauling, placing Caltech at the vanguard of the nascent discipline. The epilogue recounts the paradigm shift from protein to DNA and raises questions of continuity and change in the intellectual and social goals of post-war molecular biology. This book is not a micro-study of the American path to the double helix. On the contrary, the wrong turns have been given considerable attention in order to capture the cognitive thrust of a quarter of a century of the molecular biology program, the protein paradigm. Moreover, these chapters are not, by any means, a history of biology at Caltech. Important projects been left out, such as Dobzhansky's work in evolutionary biology and many of the activities in plant physiology and neurophysiology. The first two were peripheral to the Rockefeller program, and the third, though programmatically central, for historical reasons played only a secondary role in the rise of molecular biology at Caltech. More importantly, the narrative in this book is not the story of the rise of the new biology, as there are other possible narratives, including the versions of those bracketed out of the consensus. Rather, it is a story told mostly from the select perspective of the winners, who not only shaped historical events but, through their documentation projects, influenced the historical reconstruction of these events. Like all record repositories, the archives of the Rockefeller Foundation and the California Institute of Technology, the two principal sources informing this study, carry the inscriptions of their own historical perspectives and biases. The following pages, then, provide a particular historical reconstruction of those activities that together shaped a distinctive kind of biology centered around the physicochemical study of the gene. It is a critical study of the story of the winners, who joined intellectual and social forces, propelling Caltech to the vanguard of a new biology that came to dominate life science during the second half of the 20th century.